Hello, and welcome to today's Texas Science Festival session, Big Strides in Fighting Cancer, with Dr. Jim Allison and Dr. Livia Eberlin. We're so glad that you're all here with us today. My name is Jim Wilson. I'm the Chief Scientific Officer at the Cancer Prevention and Research Institute of Texas. Just a few notes. All participants are going to be muted without video for the duration of the webinar. And because this is one of our Science Sparks events, the organization is for each speaker to talk for about 10 minutes, followed by a Q&A. And then we plan to use the Q&A feature, ask you to hover over the bottom of the center of your screen to submit questions. We also will have pre-submitted questions that you've provided, and I'll do our best to get as many in as possible. I would now like to introduce our first speaker, Dr. Jim Allison, a CPRIT established investigator and director of the Parker Institute for Cancer Research and the executive director of Immunotherapy platform at MD Anderson Cancer Center. Dr. Allison earned the 2018 Nobel Prize in Physiology or Medicine, which he shared with Dr. Daisuke Honjo for their discovery of an effective new attack on cancer by treating the immune system rather than the tumor. And it's really a great pleasure to have Dr. Allison with us today. Jim, thank you for being here.
Hey, whoops. Thank you very much. I'm happy to have a chance to be here. Given the time, I think I'll just start. What I'd like to do today is just sort of review some of the work that's been going on and over the last 15 years, really, that's, I think, giving us new ways of treating cancer. And it's really letting people know for the first time, I think, that getting diagnosed with cancer is not necessarily a death sentence. Why would you want to, and this involves using the immune system to treat cancer. Why would you want to do that? Well, we know that cancer is caused by mutations, lots of mutations, some of which occur with time and then end up, when some of them hit a driver mutation, it's called, something that affects cell growth or its localization, allows metastases, then you develop cancer. And there are about 30,000 genes in a cancer cell that can have mutations that could conceivably cause cancer. But using the immune system rather than drugs, which either kill tumor cells by non-specific things or even by targeting those driver mutations, so-called, offers lots of advantages. First of all, there's specificity. What we know now is that T cells, the soldiers of your immune system, can recognize any mutated protein in your body, whatever it is, because peptides are put up on the cell surface. The second thing is, unlike other drugs, which as soon as you stop administering them, they're gone. Once you've got a T cell, some of them become stem-like cells that persist for the rest of your life and can be remobilized if you need them, if the tumor occurs. And finally, there's adaptability. You've probably got 100 million different T cells going through your body in small numbers of each one. But they can expand and generate an army to go out there and deal with cancer. And so the bottom line is many of us immunologists have felt for a long time that the immune system is a match for cancer. And I'll just show you one of the successes first. I was at Berkeley. We understood that this molecule CTLA-4 serves to limit immune responses and keep them from hurting you. But I had the idea that if we just could block that, and I'll show you the mechanism in a moment, we could unleash the immune system to attack cancer. So this is a patient that was diagnosed in 2004 when I was at Sloan Kettering. She was one of the first patients on one of our earliest trials. She was 22 years old, had just finished college, gotten a degree, was about ready to start her life, was engaged to be married. She had some exhaustion, some side effects. Anyway, she went to see her doctor and they diagnosed her with stage 4 metastatic melanoma. And you can see this CAT scan of her lungs. She had 31 lung metastases. She had 2 cutaneous tumors on her body, but also she had a half-centimeter brain tumor. But anyway, after a single round of treatment with our anti-CTLA-4 antibody, those tumors completely disappeared in her lungs, as you can see here, and this brain tumor also disappeared. And that was about 2006. And she's alive and well today. She's now married. She has 2 children. She's a good friend of mine now. She actually went with me to Stockholm for the Nobel Prize. And so she's 15 years out from when she first had a remission with that treatment. So this does work, and it works in a lot of people. So there she is, a photo taken 10 years after she was in remission with her two lovely children. But this is basically how it works. T cells need two signals to go, one through the T cell receptor when they recognize antigen and MHC molecules, and the other one by CD28. Tumors can't provide those, so they're invisible to the immune system until they get large enough that they die. And phagocytosis by these antigen-presenting cells, they pick up the tumor bits, redisplay them on their cell surface in the context of CD28. And that starts proliferation. But after a time, CTLA-4 comes up and turns that off. So they can't proliferate anymore, and the immune system response starts. Now, the simple idea, if you just took off the brakes transiently by putting in an antibody to CTLA-4 to keep it from interacting with the B7 molecules that they both bind to, you could stop T cell responses. And as I showed you, that works. This happens when tumors die in necrotic cell death that cause inflammation. But we felt this was compelling back in 1994 when we first started doing mouse experiments that showed this. I had the idea that this was compelling for a couple of reasons. One is, since it starts with tumor cell death, you could prime immune responses by surgical techniques, by chemotherapy, by radiation, or by these new genomically targeted agents, all of which kill tumor cells, all of which will start an immune response, but that would be stopped in its tracks by this CTLA-4 pathway. If there's continued signaling for a while, CTLA-4 tells the body that that's probably self and it ought to not attack it anymore. The immune system ought not to attack it. The second thing is, you can notice here, we're not treating the cancer. We're treating the immune cells. So this has the possibility for working, with any kind of cancer. And so for these two reasons, we managed to get it into the clinic, developed this antibody. It was approved by the FDA. Now any doctor can prescribe it since 2011. It was the first immune drug like this to be approved. And the follow-up study in 2015 shows that over 20% of the patients who received a single round of therapy with this are still alive 10 years after treatment. They're basically cured. As you can see, this survival curve goes flat at about 5 years, and nobody passes away. Virtually nobody passes away after that. So it's a very, very durable type of response. In any event, just as our phase 1 data were coming in, Tasuku Hanjo read about it and studied a molecule, showed that a molecule that he'd been studying for some time, PD-1, was another molecule like this. It works very differently, though. T cells make gamma interferon, and that makes the tumor cell put up PD-L1, which then binds to PD-1, which is also on an activated T cell. And that stops the T cell by signaling directly into the T cell, stops it from proliferating. And the cells eventually become exhausted and can't work anymore. But you can invigorate them just by taking that signal off with the antibodies. So very different mechanism. It works at the level of T cell receptor, not at CD28. In any event, these two work very well together and have been combined in metastatic melanoma. And now we know that the survival rate of metastatic melanoma patients after 5 years is almost 55%. And since CTLA-4 blockade, ipilimumab, is a part of that, that gives us confidence that this will be a durable response. And there's no reason why these shouldn't last 10 years and more. And so we've gone from, and I must point out that when Sharon was diagnosed in 2004, the median life expectancy after diagnosis was 7 months. And virtually no one, less than 2% of people, live 5 years. And now with these new treatments, with this combination treatment, 55% of patients in a fairly large study are still alive 5 years after, again, a single round of treatment with these drugs. So very effective drugs. And so it's not just melanoma anymore. Melanoma was the first. I said, anti-CTLA-4, this antibody that we made, was approved in 2011. A PD-1 antibody made by Tasuku's technique was approved in 2014 for melanoma. But now they're approved for small cell and non-small cell lung cancer, kidney cancer, colorectal cancer, head and neck cancer, some lymphomas, hepatocellular carcinoma, goes on, gastric tumors, cervical carcinoma, breast, squamous cell, goes on and on. not everything, but there are over 40 approvals now of these antibodies in this class for different kinds of cancer. And including one that's very interesting, that patients whose cancers, doesn't matter what kind of cancer they are, if they have defects in their ability to repair DNA damage, they have a lot of mutations. They give a lot of targets for the immune system. And so any patient with defects in that can be treated with these drugs now. Noticeably absent, though, are some other cancers such as glioblastoma and pancreatic cancer. Those have not as yet been very susceptible to this kind of treatment, but we're working on those now. At present, too, the response rates for other types of cancers other than melanoma are lower than that, 20 to 50%. But we've been involved with some other groups in studies in pancreatic cancer where we combine 5 different things, including chemotherapy and radiation, plus 3 different immunotherapies. We're beginning to see some responses there. So with additional work on learning more about the immunobiology of cancer, about the cells in the tumor microenvironment, because it's not just T cells in cancer, there's myeloid cells, which can inhibit T cells, or they can help them. And we're learning now the difference between those and how we can affect them. And also how we can combine conventional therapies with immunotherapy. As I told you, anything that kills a tumor cell can prime an immune response and start something that can be sustained then with immunotherapy. So the point is that now we know that the 3 traditional target kinds of therapy have been surgery, radiation, and chemotherapy. But now we can add to that immunotherapy as the 4th. And the unique thing about immunotherapy is the others can be used in combination with it. And so we're starting an institute to begin to integrate all these to try to get just a totally integrated way of using conventional and immunogenic therapies to treat cancer. So this is what we've been used to since the advent of chemotherapy and radiation in big time. Well, actually, radiation's been around since 1900. Chemotherapy starting after World War II with studies of some of the chemical warfare agents that were used in early chemos. And the idea there was just to treat large numbers of patients, look for median survival, and if you see a statistical increase, you declare it, victory. You got a month or 6 months or a year maybe. But none of these therapies have really been proven to be very curative, with few exceptions, because the tumor, with all those mutations, can find a way to escape it. We know from ipilimumab that you can move that over. In the case of Ipi, it was 4 months, but that tail in the survival curve at 20% for 10 years shows that you can cure a fraction of patients. And our goal now on this red curve isn't data; it's aspiration of what we can do by identifying new targets and using these in combination. So that's our goal now, not to just improve the median survival a bit, but to get these durable responses to actually cure cancer and cure it in as many patients as we can with as many different types of cancer as we can. And the good news is we know the basic rules now. It's just a matter of studying the individual cancers. They do differ somewhat, so it isn't quite as universal as we had hoped for any individual drug, but the combinations, I think, will help us treat at least some fraction of hopefully every kind of cancer. So with that, I'll stop, and I'll be happy to participate in the questions.
From the point of view of a medical oncologist, just recognizing just how much this new therapeutic approach has transformed our approach to almost all diseases. Let's begin with a couple of questions from our audience. The first is from Jacqueline, who asked, does this immunotherapy treatment affect patients' response to infectious diseases?
That's an excellent question. And there are toxicities associated with it, which I didn't mention. They are potentially fatal if they're not treated, but doctors have developed ways of dealing with them, but they are fairly serious. So they're not something you'd want to use with a conventional vaccine, for example. On the other hand, with some chronic virus infections like hepatitis virus infections or maybe HIV, I think they could be used to reawaken the immune system to deal with those, and those kind of efforts are just beginning, especially for chronic virus infection.
Right. So the next question, I think you've already answered, but it does open up some interesting discussions, perhaps. The question is, does immunotherapy work on advanced stage cancers? And we saw just dramatic examples. Maybe you could comment on the potential for immunotherapy to move up earlier in the management of.
Yeah, I'll answer that question. Of course, it was tested initially, as all new drugs for cancer are, on patients who have little to lose, frankly, on absolutely worse. As I said, the kind of cancer that Sharon had, her life expectancy was 7 months after diagnosis. But with everything, now it's being used earlier and earlier and earlier, and now it can be used really for early stage cancer, or indeed for cancer that was metastatic cancer that's caught early enough where the doctors think that they've removed all the metastases surgically, but immunotherapy after that's been shown to prevent recurrence. And so in both those ways, both earlier and then after surgery in patients that have minimal residual disease as well.
Right. So Cindy asks, is the long-term survival rate affected by a patient's age?
I don't know of the actual statistics on that, but I don't think very much, because I know of an 83-year-old woman who was treated. She had melanoma, and she was treated and responded quite well. And in prostate cancers, we're not doing so well there yet. It's early days, but that combination of those two drugs, we've seen some responders in people who are in their 70s, late 70s. There may be, but it's not a guarantee. It's not for sure. It's not going to work in older people. But it looks like it can. It's pretty good at rejuvenating your immune system, actually.
Jim, I wonder if, since many of in our audience are young scientists thinking about the future, do you have any thoughts regarding where the exciting applications of immunotherapy research are going to be in the future?
I think that we're doing a little bit of work in my lab now. We have some evidence for connections with the nervous system, with early stages of neuroimmunology. We're looking at the effect of that on cancer responses, but I think there's a lot of hope in neurological diseases for trying to manipulate. There's evidence that some kinds, Alzheimer's, for example, are caused in part by antibodies, by immunological responses gone awry, and I think there's a place there. But I'd like to also say that, just point out for young scientists in general, that I didn't start this work to try to figure out a way to treat cancer. I was fascinated by T cells. When I was a student at UT Austin, Bill Mandy, who's taught an immunology course there, and at the end of it, he taught antibodies. But he had one lecture right at the end of T cells. They'd just been discovered in '68 when I was a student there, and he said, these T cells, these new things, they go all over your body. They percolate in your tissues and your blood, and they go all over, looking for infections or stuff that's wrong and deal with it. And I said, gosh, how did that work? And he said, we don't have a clue. At the time, he said, I think it's just an antibody stuck on another cell, because nobody could imagine that. But anyway, I said, well, that's what I'm going to work on, because I want to understand how that works. But along the way, because I'd had a lot of family history of cancer, anytime I got somewhere where I thought we might be able to give it a little push in that direction, I took it. But the work was motivated by just basic issues, fundamental science, and how does the system works. And so there's still a big place for that. And I was lucky enough that it did work into something that was useful and made the world a little bit better place, but I hope, but I did it for the fun of discovery.
Here's another interesting question from Ellen. Does this research have any potential applications in autoimmune diseases such as lupus or MS?
Yes, what I would admit is that in our early days, we had some animal models of autoimmune diseases that we studied them because we figured if we could make them really bad, we might be able to use those lessons to treat cancer. But now we've learned the lesson. I think we're on the verge of being able to turn that around. If we could come up with some way of giving an agonistic signal through CTLA-4 in an antigen-specific way, which we can do very small scale, but the ways that we've done it so far are just not amenable to clinical application. But I think, with further study, I do believe you can. One thing out of this work that has already been applied and is still being applied is the use of a soluble form of the CTLA-4 molecule itself to bind up all those B7 molecules, which stops that second signal that T cells use. That's very useful in treating rheumatoid arthritis and some other kinds of diseases. But there are other ways, I think, of generalizing to diabetes and other things that are underway.
And I think this is going to be our final question, and it comes from Michelle, who asked, why does immunotherapy seem to work well in some people and not in others? And why does it stop working?
Well, I wish I knew the answer to that first question. I think that we're still learning. As I said, it's not as simple as these two cell interactions. There are these myeloid cells in the tumor microenvironment. Myeloid cells, their job's to repair a wound. And when they get in cancer, they try to protect the cancer and help, in a way, repair it. So they try to turn the T cells off. And so we've got to find some way to stopping them from stopping the T cells. So maybe some people, some kinds of cancer, just get a lot of those. Other than that, I don't know. I mean, I wish that we could predict who's going to respond. We just can't right now. We can, after somebody has gotten a shot, of the drug, within a few weeks, we could tell who's going to respond, but we can't predict it before they do. And what makes it stop are generally mutations in the tumor cell. For example, the T cells make two compounds, one gamma interferon, one called TNF alpha, tumor necrosis factor alpha. Those bind to receptors on the tumor cell and activate signaling pathways that make the tumor cell die. But it's been shown now that a lot of melanoma patients that escape this therapy have mutations in the signaling pathway for gamma interferon. So the lytic signal from the T cell just doesn't work anymore. So there's ways around that we're working on, but it's this constant balance because of all the mutations in the tumor cell, and gives it a lot of substrate, if you will, for evolution, for selection to work on. Take out all the cells that are susceptible. There's always going to be one there with something else, and luckily the immune system can change as well. So that's what we're trying to do, is keep up with that evolution of the tumor.
Dr. Jim Allison, thank you for being with us. The story of your contributions and also your excitement about medical research is just inspiring all of us.
Good.
Thank everyone for their questions, and we're now, I think, have the opportunity to hear from our next speaker, Dr. Livia Eberlin. Dr. Eberlin is assistant professor in the Department of Chemistry at UT Austin. Dr. Eberlin is the inventor of the MasSpec Pen, a device that can detect cancer during surgery in real time. Her innovation has been recognized with several prestigious prizes, including the MacArthur Genius Fellowship and the 2020 Eli Young Investigator Award in Analytical Chemistry. Dr. Eberlin, welcome.
Thank you. Okay, so I am incredibly honored to be speaking to you today and sharing the research that my lab has been pursuing, developing the MasSpec Pen technology, and how this technology, this device, has really allowed us to bridge science and medicine with the intent of improving patient care, especially care for cancer patients. So I'm sure many of you that are here today have had the heartbreaking experience of a cancer diagnosis, either through family members or friends, or even yourself as a patient. And as Dr. Allison said in his presentation, cancer surgery remains as one of the main treatment options for patients with cancer. And despite the improvements and advances in chemotherapy and immunotherapy, most patients that present a solid tumor will be subjected to a cancer surgery procedure. And you may realize that one of the main questions that patients ask coming out of a surgery is, was the surgeon able to remove it all? Am I cured? Is all of this disease, cancer tissue out of my body? And to be able to answer this question, we really need to recognize how complex cancer surgeries can actually be. And even though surgeons are incredibly talented and trained and experts at recognizing tumor visually, right, by gross anatomy, actually looking at these organs, it can be very tricky to identify tumor, especially in the boundaries between the tumor mass and the adjacent normal tissue that is surrounding the cancer tissue. And you may imagine that removing all of this tumor, there's actually significant clinical data that shows that getting all of the tumor out in the first surgical procedure really improves its chances of survival for patients. So the way that surgeons actually verify if all the cancer tissue has been removed is through what we call ex vivo frozen section analysis with the assistance of pathologists. And just to give you an idea, this process has been in place in clinical practice for now over 100 years. So this tissue that is removed from the patient is sent to an adjacent room where pathologists will freeze, section, and look under a light microscope and try to identify, looking at the boundaries of the tissue, if there's any cancer remaining at that boundary or not. And then they communicate those results to the surgeons that are in the operating room. Now, this process has been incredibly powerful to advance care for cancer patients and to enable surgeons to more precisely identify the status of the margins of this tumor resection, but it also has many problems. It's a time-consuming process. Sometimes it can take up to 30 to 45 minutes for frozen section analysis while the patient is still in the surgical bed. And it can be inaccurate because of this really quick processing that the tissue is subjected to. For pancreatic cancer surgeries, for example, there's clinical data showing that in certain sites, you can see up to 10 to 25, 27% error in surgical margin evaluation with histopathology. For breast cancer patients, there's also extensive clinical data showing that about 20 to 25% of patients will come back for a second surgery because not all of the tumor was removed in this first surgical procedure. So I think I can make an argument here that there is an incredible need for new technologies that can ideally be implemented in the operating room to help surgeons in this surgical process of making a decision and also surgical margin evaluation. And even more ideally, it would be incredible if the technology allowed in vivo analysis of the tissue in a way that is non-destructive and safe for the patients. So with that in mind, my lab, which we're majorly composed of chemists, developed what we call the MasSpec Pen device or the MasSpec Pen technology. So the MasSpec Pen is a handheld tool that we actually 3D print in my lab. Very easy to use, that is connected to a mass spectrometer. And the entire process is triggered with the click of a foot pedal. So the device, once the surgeon actually touches the tissue, we get to perform one of the simplest and most beautiful, I would say, chemical experiments, which is a liquid extraction. So we actually pump one droplet of water, in this case, as our solvent, onto the tissue surface. And water, as we know, is an incredible solvent. And water very gently extracts molecules that are natural on the tissue into the droplet. And then once we have these molecules that we know are very characteristic of tissue into our droplet system, we use a vacuum to transport this droplet from the tip of the pen all the way to our mass spectrometer through this tubing system. And once this droplet reaches the mass spec, we actually get these beautiful chemical profiles that show the abundance of molecules, of the molecules that were extracted from the tissue. When we design the MasSpec Pen, we really want to make it easy to use and intuitive to the surgeons. So one can unplug the device can be sterilized, it can be disposed or reused. So there's a lot of flexibility in what you can do with the technology. Let me cancel this call. Okay. So once we get this really complex molecular data, it's pretty powerful to see the amount of molecules that we can actually detect. We detect metabolites in our mass spectrum, and that's the data we're getting from the mass spectrometer. We detect a variety of fatty acids and lipid species that we know from extensive research that's been done by many investigators are very characteristic of disease state of cancer tissue or a normal tissue and even subtypes of cancer. Now, to kind of deconvolute this really complex molecular data, we use machine learning and artificial intelligence algorithms to go from this complex molecular data to an actual clinical decision or something that could inform a clinical decision. And we're able to tell, based on this profile of molecules, if tissue that was analyzed is normal or cancerous. So we've now applied the MasSpec Pen in my lab in really over, I think, 1,200 tissues in the lab to develop statistical classifiers with machine learning algorithms. As I said, this extraction is extremely gentle, so the analysis is non-destructive. The entire analysis is also really fast, which is one thing that we really wanted to achieve for clinical use. It's under 10 seconds. And with our classifiers, we've kept an accuracy about 96% for cancer diagnosis. As you can see, we've explored a variety of different tumors. We've also been able to subtype tumors. So not only say if it's normal or cancerous, but different types of tumors have different molecular profiles. So we can pick that up with mass spectrometry, and even in some cases pinpoint genetic mutations. In the case of gliomas, we can identify an IDH1 mutation that has really implications in treatment options for the patient. So one of the incredible things about Texas is that not only we're able to do incredible science and advanced technology, but Texas, the state of Texas, gives an opportunity to translate technology and explore these applications in medicine in really a unique way. So we've transferred our system from UT Austin to Baylor College of Medicine, and we also now have a system being installed at MD Anderson to actually start our pilot clinical studies. Before I show you those results of actual life, I'd like to show you art imitating life in an episode of Grey's Anatomy.
It really works.
Oops. Okay, there you go.
Supposed to, it's still in the testing stage. We're testing it on this guy. He's got a wife and 2 kids. They're very nice, and they really, really want him to stay alive. And I promised them I'd make sure he gets the best possible care. So maybe, Dr. Gray, if you could just tell
Schmidt,
Dr. Wilson.
be quiet. She's got it. Look, the distal end, adenocarcinoma. And if I go 1 centimeter approximately.
It works. It's working.
I'm gonna save half his stomach. Now you get to go tell his family that not only did you save his life, you saved his quality of life. I'm gonna go check on our stoners.
Okay, so we're actually really pleased how well, the TV show, did in picturing our technology. But of course, as scientists, we wanted to test it ourselves. So we set up our real life Grey's Anatomy or our pilot clinical study in Houston. This was actually our very first surgical procedure. This is me here holding our version 2 of the MasSpec Pen with our interface in burnt orange that we installed in the operating room. This here in the middle is Dr. James Sulliberg. He was actually the first surgeon to use the MasSpec Pen. Now we've had over, I think, 8 different surgeons that have used the technology. I'm testing it in surgical procedures. So this is actually our system installed in the operating room. Here again is Dr. Sulliberg. He's using the MasSpec Pen here in a thyroid cancer procedure. So mostly for just verifying the diagnosis of the tissue at the time of surgical resection. And you can see here a little bit the pan. This is the mass spectrometer and the interface. We're now moving the technology towards a mobile system that can get much closer to the patient. It's much more simplified and easy to use in terms of the mass spectrometer system, so that we can allow for using multiple operating rooms and just getting closer to the patient as well for the analysis that needs to be done. This is an actual video of the MasSpec Pen being used in vivo, so while the patient is under anesthesia. So you can see here Dr. Sulliberg doing analysis of a thyroid, and then in real time, nearly real time, we get the molecular data here being acquired in the mass spectrometer. Here's just a closer picture of the use of the MasSpec Pen. So through our pilot clinical studies, we have really understood some of the power of this technology in the aspect of cancer surgeries. We've also continued to advance and refine our tools and make changes so that we can more broadly use in a variety of surgical procedures. So far, we've used the MasSpec Pen in almost 150 surgeries, mostly breast, pancreatic cancer, parathyroid, and thyroid procedures. One thing that's been really incredible is that there have been no limitations to tissue analysis or diagnosis due to the use of the MasSpec Pen. So that chemical extraction is very gentle to the tissue. You couldn't even recognize that the tissue has been analyzed, which I would argue is extremely powerful when you're trying to incorporate a new technology into a clinical workflow, like a complex workflow like a surgery. Also, very importantly, we haven't seen or reported any complications or adverse effects of the MasSpec Pen. They're all sterilized, safe to use in the patients, and all of the materials are biocompatible as well. For pancreatic cancer, these are our pilot results. We're still refining the technology. We have about 94% accuracy for in vivo cancer detection and margin evaluation. And for breast cancer, we're about 95%. And continue to increase our numbers here in terms of patients so that we can more rigorously evaluate the performance of the technology. So to summarize, we've developed really a user-friendly platform through a simplified device that incorporates some basic principles of chemical analysis into what we call a medical device that can be employed in the clinic. We're able to perform molecular analysis nearly in near time and probe this molecular composition of the tissue in order to be able to provide a diagnosis. The MasSpec Pen is biocompatible, the process is automated, it's disposable and very non-destructive to the tissue. And one of the things is it can be adapted to many surgical modalities. We actually have a study where we tested the MasSpec Pen in a robotics procedure and got their in vivo results from that as well. So ultimately, we believe the MasSpec Pen can really redefine how surgery is performed. We hope to be able to improve accuracy in diagnosis, precision in surgical resections, but also expedite these procedures in the surgical procedure for the patient, that surgical treatment, by enabling surgeons to get their real-time feedback. And ultimately, we could reduce healthcare costs and most importantly, reduce the rate of second surgeries to improve treatment and outcome for patients. So I just want to quickly thank my incredible research team. This was our last picture together. This is our current Zoom picture. They have been so passionate and involved in this development. Also want to thank all of my incredible clinical collaborators that, have taken the risk in testing a new technology. And it's just been a really exciting project at Baylor College of Medicine and MD Anderson especially. In funding, I'm incredibly thankful to CPRIT for many grants that we have received to support this and related research, as well as the NIH and the Moore Foundation especially as well for their support. So with that, I'll be happy to take any questions.
Wonderful, Dr. Eberlin. Thank you so much. We do have questions, and the first comes from Aaron, who asks, can the MasSpec Pen distinguish between different tissues or only between cancer and non-cancer cells?
Oh, that's a great question. No, it can distinguish between different tissues. We have used the MasSpec Pen to distinguish, for example, in benign parathyroid procedures, a parathyroid can very much resemble a lymph node. So we can very easily distinguish two different organs. We can distinguish even different cell types. We can distinguish epithelial cells from stroma cells, necrotic cells, fat cells, and so on. We also have a project in endometriosis where we can distinguish the endometriosis lesion from the adjacent tissues that the lesions may be within. So actually distinguishing tissue types is very, very obvious from our molecular data, distinguishing cancer from normal as well. And as I said, for many cases, we can also distinguish histologic subtypes of cancers. And when there's an oncometabolite that is indicative of a mutation, we can actually also pick them up.
The next question is from Carla, who asks, do you foresee being able to use this technology during endoscopy or laparoscopic procedures?
Yeah, absolutely. So we worked with Intuitive Surgical in a collaboration also with Thermo Fisher Scientific, and we developed a drop-in version of the MasSpec Pen. So it's not handheld, it's a lot smaller, but it has a fin that the surgical robot can grasp, or if you have a laparoscopic kind of grasper, and then you can use it in vivo in a laparoscopic or a robotic procedure. So we tested that in a surgery with a pig, and we got in vivo data for different tissue types showing that the technology works under kind of like the laparoscopic environment, which, there's an atmosphere of CO2, right, that is used to inflate the abdomen, and we're able to get molecular data from that as well. So we're moving forward with that for robotic prostatectomy procedures.
Wow. Tina asks, when do you think we'll be able to implement the use of the pen worldwide?
Well, that's really mostly a regulatory question. So medical device, of course, I'm very excited about technology and, especially technologies that come from academic institutions. I think it can be really exciting, which the MasSpec Pen is, but then we do have to respect the regulatory process, of course, to get a technology actually into a medical device. So that's really the more time-consuming step. It's getting to a final design of the system and then going through all of the FDA requirements, which are very much necessary to show that the device is really the efficacy and it's safe for use in humans. So that's the process. So it does take a few years.
And Arthur, I asked the questions a bit of a follow-up, wondering about the affordability of the pen for perhaps smaller hospitals and wider dissemination.
Yeah, that's a great question. So the pen itself is extremely cheap. We 3D print them in my lab. We have two 3D printers that we use for prototyping, and it's just resins. The pen tip, I failed to say that, that's made of a PDMS, polydimethylsiloxane. That's a biocompatible polymer. It's FDA approved for medical implants, so very safe for use in humans. And then everything else is just plastic. So we made it that way, so we wanted to be disposable. So that's relatively cheap. Now let's talk about mass spectrometers, right? That is the costly part of the equations. And you'd be surprised how many hospitals are already suited with mass spectrometry technology. You know, a lot of clinical laboratories use MasSpec for diagnostic assays. But what we envision is like a smaller version of the mass spectrometer that's kind of like a roll-in unit that can go in and out of surgeries. And that would be like a one-time kind of system that would be acquired by the hospital, and then it could be reused in many procedures. And if you compare with like PET scans or MRI scans, I think, you know, the price actually would be lower than that, but that's something that still needs to be worked on.
Doug asked an interesting question, wondering about the MasSpec replacing genomic profiling of tumors.
Oh, I don't think there's a replacing here in discussion. I don't think anything will replace genomic profiling. Is extremely powerful for pinpointing mutations with really high specificity. It's really the perspective of use here is different. It's really the how quick it is that we can enable acquisition of molecular information. And remember, genomic profiling, we're looking at genes, right? And we're using here metabolites, so small molecules. So it's actually complementary information. I think, you know, MasSpec can complement what genetic testing does. But I don't think it's a replacement question here. Is really more technology to improve patients.
Livia, one of the inspiring parts of your story is how engaged you are with clinical problems and clinicians. How does a chemist get to know important medical problems and actually develop the colleagues that are enabling the translation?
Yeah, that's such a great question. So I've always been passionate about medical research, but my first passion is mass spectrometry. So I couldn't get away from that. But during my PhD, I had the opportunity to go to the hospitals to collect samples, which at that time seemed like a very labor-intensive kind of job. But through that process, I actually just was hanging out in hospitals, and I was watching, you know, surgeons, pathologists working and making these clinical decisions. And at that point, I recognized, wow, we have this incredible science and technology that we develop in like academic institutions, but they're mostly like R&D kind of technology. They're too complex, right? Like it's really complex to give a mass spectrometer to a clinician. And then through conversations with colleagues and surgeons, and just recognize, okay, what are the features, device features that we need to actually make this usable? So I really, you know, enjoyed working with clinicians. That's part of my research program, and having that interaction is really crucial to get their feedback on what's actually useful. Because I think as scientists, we can get really deep into the science, and then recognizing that, well, we need feedback from the actual users, right? Our colleagues that are the clinicians seeing patients to actually implement it. That was really crucial.
Well, I think that those final comments are a really wonderful way of capping these two presentations of how science is impacting on real important problems in medicine, but also the excitement of that journey from discovery into patients. Dr. Eberlin and Dr. Allison, thank you for joining us today. I want to thank all of those who are participating in your questions, and ask you to be sure to visit the science.utexas.edu to sign up for more sessions between now and March 26. And to contact the College of Natural Sciences at cnsdev@austin.utexas.edu if you have follow-up from today's session. Thank you for your interest in Texas science, and we hope to see you virtually at more sessions.